Hello children, welcome to NEET AIPMT Foundation. This is biology session. You are watching sexual reproduction in plants part 2. Children, in the yesterday's class we have a brief introduction and about the male reproductive organ, that is stamen, and the formation of male gametes. What do you call the male gamete? Microspore. Okay. The formation of male gametes is called microsporogenesis. Okay. So, this is the stamens, the male reproductive part is called anther. So, this anther consisting of lobes, okay, it is diathecous. Okay, there are 4 lobes are present, they are called pollen sacs or microsporangia. Okay. And we studied about the structure of pollen grain, etc., how the pollen grain is formed. Okay, this is the pollen grain. So, as soon as it reaches the stigma of the flower, it starts germinating and produce the tube, pollen tube, and involves in the process of fertilization. So, let us continue today. Are you ready, children? Development of female gametophyte. Development of female gametophyte. The female reproductive organ is called pistil. A pistil has a terminal receptive disc-like stigma, a stalk-like style, and a basal swollen ovule-bearing part called ovary is present. Inside the ovary is the ovarian cavity with one or more locules and the parenchymatous cushion called placenta, from which arise ovules. Okay, how the ovules are formed, that we are going to discuss. A typical pistil consisting of the basal swollen part that is called ovary, and a stalk called style, and a terminal receptive disc, it is called as a stigma. Okay. Inside the ovary there are one or more ovules, or they are also called megasporangia. Megasporangia. The ovule is an integumented megasporangium found in spermatophytes which develop into seeds after fertilization. Okay. The main body of the ovule, consists of parenchymatous tissue, the nucellus, with one or two coverings, they are called integuments. Okay. It consists of parenchymatous tissue, nucellus, that is with one or two coverings, that is called integuments. Okay. The integument surrounds the nucellus. Integuments surround nucellus all around except the apex, leaving a narrow passage, it is called a micropyle. Okay. Micropyle, we can also call it as a opening of the ovule. Okay. Each ovule is attached to the placenta by a slender stalk that is called funicle. The point of attachment of the body of the ovule with the funicle, it is known as hilum. Okay. The ovule at first arises as a primordium on the placenta in the cavity of ovary. Okay. So, here you can see this is the pistil, this entire thing. Okay. It's pollen receptacle, pollen tube. As soon as the pollen grain reach this stigma, the center one is called a pistil. It consists of 3 parts, that is stigma, style, and this part is called the main part, the swollen part, it is called ovary. Okay. Inside the ovary, ovules are present. So, these ovules only develop into the embryo sac, the female gametophyte. Okay. Owing to the meristematic activity of the cells of ovular primordial, the protuberances, the outgrowths become prominent and constitute the nucellus. Okay. The initials of the two integuments arise at the base of the nucellus and surround it all around except the apex. The process of Megasporogenesis begins with the differentiation of a primary archisporial cell in the nucellar hypodermis. Okay, it starts in the nucellar hypodermis. The primary archisporial cell divides to form the outer primary cell wall, and inner behaves as a megaspore mother cell. Once again, MMC. Their microspore mother cell, here we are calling it as a megaspore mother cell, and give rise to 4 potential megaspores by meiosis. Okay, therefore microspores, here 4 megaspores. Okay. Next, the 4 megaspores are arranged in a linear tetrad. Usually, one megaspore of the tetrad becomes functional and develops further, remaining 3 will degenerate. Okay. The functional megaspore is first cell of the female gametophyte. It divides by 3 successive divisions to form an 8 nucleate female gametophyte. It is also called as a embryo sac. Okay. First 4 megaspores are formed. From that 4 megaspores, 3 will degenerate and disappears, and 1 megaspore, it is a functional megaspore mother cell which divide by 3 successive divisions to form an 8-celled, 8 nucleate female gametophyte, that is called as a embryo sac. The 3 of the 4 nucleate or the 3 of the 4 nuclei nearest to the micropyle. Micropyle means the opening of the ovule, the opening in the ovule through which the pollen tube will enter, the differentiate into the egg apparatus. Okay. The 3 of the 4 nuclei nearest to the micropyle differentiate into egg apparatus which contain the egg. So, here once again I am repeating, each ovule has a 1 or 2 protective envelopes called integuments. The integument encircle the integuments, 2 integuments are there, the integuments encircle the ovule except at the tip where a small opening present, that is called micropyle. Then the opposite the micropylar end there is a chalaza representing the basal part of the ovule. So, enclosed within the integuments there is a mass of cells called nucellus. The cells of nucellus have abundant reserve food materials, and the located in the nucellus is the embryo sac. This embryo sac is called as a female gametophyte. An ovule generally has a single embryo sac formed from a functional megaspore. Okay. The process of formation of megaspores from the megaspore mother cell is called as a Megasporogenesis. Megasporogenesis. Ovules generally differentiate in a single megaspore mother cell, that is MMC, in the micropylar region of the nucellus. The megaspore mother cell undergoes meiosis division to form 4 haploid megaspores. Out of 4 megaspores in a linear tetrad, usually the upper 3 degenerate and the lowermost enlarges to form a functional megagametophyte or functional megaspore. The functional megaspore is the first, okay, the functional megaspore is first cell of the female gametophyte. The nucleus of the megaspore divides into 2, 4 and finally 8 daughter nuclei. Okay. So, here as I told you that there are 3 of the 4 nuclei nearest to the micropyle, okay, will differentiate into egg apparatus. Before that, what happened? This functional megaspore it divides by 3 successive divisions I said. So, how those 3 successive divisions takes place? First 2, then 4 and finally 8 daughter nuclei are formed by 3 successive divisions. The 4 of which are located at each pole. One nucleus from each pole migrates to the center of to form 2 polar nuclei. Okay, means further development. Out of those 8 nuclei, 2 polar nuclei, a diploid secondary nucleus, okay, and 3 nuclei at the chalazal end of the embryo sac. They are called as antipodals. Okay, see here. So, this is a typical ovule. It is having the cushion like structure placenta. On the placenta, the stalk of the ovule, it is called as funicle. Okay, the body of the ovule is called chalaza. The tissue present in the ovule, it is called nucellus. Okay, and this is the embryo sac middle. In this 8 cells are present. Okay, 3 antipodals, 3 synergids and 1 egg cell, and another one secondary nucleus. Okay, there are 2 integuments, outer integument and inner integument. Okay, this is the opening of the ovule, that is called as a micropyle. Okay, so this is the embryo sac. Okay, so in this embryo sac, one nucleus from each pole migrates to the center of the ovule or center of the embryo sac to form 2 polar nuclei, which further fuse to form a diploid secondary nucleus. These are the polar nuclei. It will form the diploid secondary nucleus. This is the secondary nucleus. Okay, it merges to form a diploid secondary nucleus. The 3 nuclei at the chalazal, this is the chalaza. The 3 nuclei at the chalazal end of the embryo sac form antipodals. These are the antipodals. Okay. Next, the remaining 3 nuclei at the micropolar end, this is the micropolar end, will get surrounded by the cytoplasm to form pyriform cells. These pyriform cells itself, these 3 cells together constitute the egg apparatus. Okay, these 3 cells, which consist of 2 cells known as synergids. These are synergids, or it is also called as helper cells. Synergids are also called as helper cells. Okay, and an egg or oosphere. See, this is the filiform apparatus. And which consists of 3 cells I said, no, there are 2 synergids, they are called helper cells, and 1 egg cell. The synergids main function is they help the pollen tube. After the entry of the pollen tube into this micropolar region, they locate or they help this pollen tube to reach the egg cell. Okay, that is why they are called helper cells. Okay, the synergids have special cellular thickenings at the micropolar tip called filiform apparatus, which play an important role in guiding the pollen tube towards the synergids. The egg cell on fertilization gives rise to zygote, while the synergids get disorganized soon after fertilization. Okay. Let us move further. The 3 of the 4 at the opposite end of the developing embryo sac, which is called the chalazal pole, they are called antipodal cells. The 2 remaining nuclei, one from each other, from each pole, migrate to the middle center of the embryo sac and form a secondary nucleus, a diploid secondary nucleus. So, this monosporic type of development generally referred to as the Polygonum type, the ovule type. It is called Polygonum type because it was first found in the Polygonium species. Okay. So, about 70% of the angiosperms, flowering plants, have this type of development only. Okay. So, this is the Polygonium type development and this is the ovule. Okay. So, this the two nuclei move to the center to form the polar nucleus, and the 8 cells present in this embryo sac, 3 antipodals which are present towards the chalaza region, and 3 cells differentiate to synergids and an egg cell present towards the micropolar region, forming the filiform apparatus. Okay. Next, types of ovules. It's very interesting to know to study the different types of ovules. Okay, before going to the types of ovule, what happened? The fertilization finished to form the zygote, while the synergids will disorganize. The antipodal cells sooner or later also get disorganized. They may, however, be nutritive in function. The antipodals helps in the nutrition of the developing embryo. Thus, the typical embryo sac contains 8 nuclei, but 7 cells, that is 3 micropylar, 3 chalazal and 1 central. Okay. The types of ovules based on the relative position of funicle. Funicle, what I said, funicle is the stalk of the ovule. So, based on the relative position of funicle, chalaza and the micropyle. Chalaza is called the body of the ovule, micropyle is called the opening of the ovule. Funicle, chalaza and micropyle position and the curvature of the embryo sac. The different types of ovules are found in angiosperms. Okay. What are they? The first one is erect type of ovule, that is also called as orthotropous ovule. Orthotropous. Okay. Erect. Depending upon the configuration and orientation of the body of ovule in relation to the funiculus, there are 6 types of ovules are there actually. What are they? Orthotropous, anatropous, hemitropous, campylotropous, amphitropous and circinotropous. Okay. So, in orthotropous ovule, the body of the ovule lies straight and upright over the funicle. Hilum, chalaza and micropyle, they occur on the same line. This type of ovule is the Polygonum type. Okay. Next, anatropous ovule, it is a inverted ovule. The body of the ovule is inverted and gets fused with the funiculus along its whole length on one side. The fused funiculus forms a ridge called raphe. Okay, the hilum and micropyle are nearby with chalaza on the opposite side. So, it is the most common type of ovule, anatropous ovule. Okay, it is an inverted ovule. Orthotropous ovule, it is erect ovule. This is the orthotropous ovule. See the position of this funicle, chalaza and micropyle. Okay. Here you can see chalaza, this is the funicle and micropyle. This is anatropous ovule. This is the most common type of ovule. Okay. Next type, amphitropous ovule. Amphitropous ovule, both the body of the ovule and the embryo sac are curved. Example, papaver and lemna. So, this is the amphitropous, both are curved. Then campylotropous, the body of the ovule is curved by the embryo sac straight. Hilum, chalaza and micropyle come nearby. Example, capsella caparis. So, this is the campylotropous. See here hilum, chalaza and micropyle come close, very near. Then the hemitropous ovule, hemianatropous, it is also called as a hemitropous, it is a half inverted ovule. Hemitropous ovule is a half inverted ovule. It is intermediate between the orthotropous and anatropous type of ovules, with the body of the ovule lying at right angles to funiculus, fusing with the latter for less than half length. Hilum, chalaza and micropyle are separate, with chalaza and micropyle being on the same straight line. Example, ranunculus. See, this is a half inverted ovule, hemitropous ovule. Okay. The next campylotropous ovule. So, campylotropous ovule, if you take the body curved, amphitropous ovule, both body and embryo sac are curved, and cicinotropous ovule, another type, that is funiculus coiled around the ovule. In a typical anatropous ovule, the funiculus is fused with the body of the ovule lengthwise beyond the hilum. It gives rise to longitudinal ridge called raphe. Okay. So, we have studied already the funiculus contains a vascular strand for the supply of nourishment of the ovule. Now, come to the pollination. What is pollination? The term pollination refers to the process of transfer of pollen grains from anther and their deposition on the stigmatic surface of the flower, or we can also define the term pollination refers to the process of transfer of pollen grains from anther of the stamen to the receptive stigma of the carpel of the same or genetically different flowers of the same species. Okay. It is called pollination. Simply transfer of pollen grains is called pollination. It is of two types, that is self pollination and cross pollination. So, what is self pollination? It is a process of transfer of pollen grains from anther to the stigma of the same flower. It is seen in the flowers like pea, wheat, rice, most of the cells and pulses. This type of pollination is observed. It is also referred to as autogamy. It is also referred to as autogamy. Autogamy is a type of self pollination in which an intersexual or perfect flower is pollinated by its own pollen. Okay, it involves the transfer of pollen grains from anther to the stigma of the same flower. So, another type is also there, that is called as gnetogamy. Gnetogamy means it is the transfer of pollen grains from the anther to the stigma of different flowers present in the same inflorescence or on the same plant. Gnetogamy is genetically equivalent to self pollination, but ecologically it is cross pollination. Okay. Autogamy and gnetogamy, these are the two types of self pollination. Okay. So, this is the self pollination. You can see this is the anther where the pollen grains are produced. If the pollen transfer in the same within the same flower, it is called as self pollination. Okay. If the pollen transfer occurs from the anther to the stigma of different flowers, then it is called cross pollination. Okay. We will discuss more about this pollination in the next class. Okay. Hope you understood, children. Read the book. Thank you for watching our video. Have a good day.